Assalamu alaikum. Hello, my dear students. How are you all? Welcome to another biology class. I'm your biology teacher, Imrijaitul Hassan. And today we are going to learn from the chapter Human Immunity. And today our lecture topics are the components of human immune systems. Um, so, our immune systems actually, it has a huge arsenal of cells. Okay. It has so many types of components like lymphocytes, phagocytes, granulocytes, blood platelets, dendritic cells, so many things. Okay, and they have to work effectively. Most of the immune cells, they need cooperation of their fellows. Okay, sometimes immune cells, they communicate by directing physical contact, sometimes by releasing chemical messengers. In order to have room for all the cells, they needed to match millions of possible enemies. The immune system stores just a few of each kind. Okay. When an antigen appears, those few matching cells multiply into a full scale of army. Okay. And after their job is done, they fade away. Okay. This is the strategy. Okay. So, our human immune cells, it includes lymphocytes and so many things. Okay, let's talk about one by one. The first one, the lymphocytes. Lymphocytes are one of the main types of immune cells. One of the main and one of the vital types of immune cells. There are different types of lymphocytes like B lymphocytes, T lymphocytes, even natural killer cells. They are also the lymphocytes. Uh, so the first one, let's talk about the first one, the B lymphocytes, which is also known as the B cells, or the cells which can produce antibodies. Okay, so these cells actually, it works chiefly by secreting soluble substances called the antibodies into the body's fluids. So antibodies, they ambush antigens. Here the antigen means any kind of foreign particles. It can be the chemical substances, it can be virus, bacteria, fungi, whatever. Anything which is not mine is called the foreign invaders. So antibodies actually, it ambush antigens circulating in the bloodstream. And each B lymphocyte, that means each B cell, is programmed to make one specific antibody. So for every individual, and so for every individual type of antibody, there is a specific B cells. Okay. When a B lymphocyte encounter its triggering antigen, it gives rise to many large cells known as plasma cells. And every plasma cells is essentially a factory for producing those specific antibodies. Each of the plasma cells descended from a given B lymphocyte manufactures millions of identical antibody molecules and pours them into the bloodstream. Okay, this is the scenario of the B lymphocytes. So you have to understand and then memorize these informations. Okay, so now the second type of lymphocytes, which is called the T lymphocytes or T cells. Okay, and these cells actually contribute to the immune defense in two major ways. The first one, some of the T lymphocytes, T cells, they direct and regulate the immune response, and others, T lymphocytes or T cells, they are the killer cells that attack cells that are infected or cancerous. Okay, so there are two types of T cells. The first type, they regulate the immune response, and the second type, they actually the killer cells, they kill the infected cells or the cancerous cells. So T lymphocytes actually, they work primarily by secreting potent chemical messengers known as cytokines. In our last lecture, we have already discussed about the cytokines. So, and more specifically, lymphokines. Actually, these T lymphocytes, more specifically, they produce the lymphokines, and these things actually, this bind to the target cells, and these lymphokines mobilize many other cells and substances. Okay, and thus by mobilizing those target cells, they encourage the growth of they also encourage the phagocytosis. Okay, phagocytosis. And another type of T cells which is called the helper T cells, and they are the subset of T cells, and these helper T cells actually they express a unique antigen on their surface called CD4 plus. That's why they actually, see, according to its name, from its name you can guess the function of the helper T cells. Actually they help the T cells, and they also helps those phagocytic cells to identify those foreign invaders. Okay. Okay, see here the another type where I have already mentioned the natural killer cells. I think about natural killer cells, these are the kind of lethal white cells or lymphocytes which are able to kill those invaders. Okay, actually I have already discussed about these natural killer cells in our previous lecture. So if you have any confusions, just review the last lecture. Now I'm going to talk about the most interesting mechanism of our immune system, and the most vital thing. Actually, this is the main immune function, immune capability, or immune system in our body, which is known as the phagocytosis. And the cells which are able to phagocyte is phagocyte, which are actually which perform this phagocytosis process, is called the phagocytes. Here the cytes mean cells, and phago, it comes from the word to eat. It means to eat. Okay, so the phagocytes are the large white cells that can swallow or digest microbes and other foreign particles. So usually the monocytes are the phagocytes, and the monocytes here, the monocytes are the type of white blood cell, you know, so that circulate in the blood. And when monocytes migrate into tissue, they develop into macrophages or big eaters. Okay, like, you know, in our country's defense systems, we have different types of defensive organization, like army, navy, air force. So how actually how a civilian they become a army, they become a navy person? How? Because the procedure is a civilian, it turns into army person when he or she goes through a rigorous training. So see here, these are the white blood cells. When they migrate into tissues where the infection occurs, and then they transform into phagocyte cells, which are called the macrophages or big eaters. Okay, so these are the specialized types of and there are also different types of specialized macrophages can be found in many organs, including the lungs, kidneys, the brains, and the livers. Some macrophages, some phagocytic cells, they remain stuck at specific organs, at specific tissues, and there are some macrophages or phagocytic cells who those cells actually they circulate through the body, circulate to the whole over the body through blood or body fluids or limbs. Okay, and by circulating they actually they act as an scavenger. Okay, they scavenge, they consume, they actually digest all the worn out cells, all the debris, all the invaders, all the foreign particles. Okay, they are the cleaner of our body, and mm, thus they actually helps us, thus they actually protects us from all those foreign particles. So these macrophages, they plays a very vital and important role by scavenging those foreign things, foreign invaders, bacteria, fungi. Okay, so see here, this is a total phagocytic process. So how this phagocytic process actually takes place? Okay, the first thing, See, this one, this is this yellowish, this is the macrophage, this is the phagocytic cell. That means this is the white blood cell, modified white blood cell, which is able to digest the foreign particles or microbes or foreign invaders. See, suppose this one here, this brownish, this component, this is the microbe or other particle, foreign invaders, any kind of foreign invaders, suppose. Okay, so and this one is the macrophage or phagocytic cell. Okay, so first thing, when the chemotaxis, at the very beginning, chemotaxis and adherence of microbes occurs. Okay, chemotaxis occurs. So, you know, what is chemotaxis? Chemotaxis means the movement uh, towards something according to its chemicals secretion or chemicals chemicals. Okay, so so chemotaxis means the movement. Taxis here, taxis means movement and chemo means chemicals. So movements according to or attracted to chemicals is called the chemotaxis. Okay, so here okay, now I think total phagocytic process, we can divide them into several phases. So at the very first, the delivery of phagocytic cells to the site of infection takes place. Okay, so the phagocytic cells, that means the monocytes or neutrophils or the macrophage cells. Okay, they actually they actually go to the target place, go to the the place where the infection occurs by two processes. Okay, the first one is the diapedesis. Okay, diapedesis is a kind of migration of cells across vascular walls, which is initiated by the mediators of the inflammations like kinins, histamines, prostaglandins. So yeah, these are the mediators, these are the inflammation mediators. When infection occurs, these mediators, they encourage the inflammation, and then when an inflammation occurs, inflammation takes place in our body, within the body, then those phagocytic cells, these phagocytic cells, they can recognize those areas. Okay, and then as a result, they can easily recognize those places and go to the target region. And another way is the chemotaxis. So the actually the eventual concentration of phagocytes at a site of injury results from this chemotactic response by these phagocytes. Okay, a number of chemotactic factors that attracts those macrophages actually secreted by these microbe foreign particles. Okay, and which is and by these for chemicals, these phagocytes are attracted. And the second phase of the phagocytic process is the phagocytic adherence. Okay, to engulf or digest these microbes, these phagocytic cells, they have to adhere those microbes. Okay, so phagocytosis actually is initiated by the adherence of a particle to the surface of the plasma membrane of a phagocyte. Okay, and this step usually involves several types of surface receptors on the phagocytic membrane. Here there are several surface receptors. Through these receptors, these phagocytic cells and these microbes can adhere with each other, and then the ingestion occurs. Okay, after the attachment of the phagocyte to its target, some sort of signal generation take place, and which is poorly understood and results in physical or chemical changes in the cell, and that triggers the ingestion. Okay, ingestion is an engulfment process that involves enfolding or invagination of the cell membrane at enclosing the particle, and ultimately releasing it into the cytoplasm of the cell within a membrane vesicle. And the end result of ingestion is entry the particle enclosed in a vesicle derived from the plasma membrane of the cell. This structure is called the phagosome. So see here the phagosome, phagocytic vesicle, And so this vesicle, engulfs those microbes or particles. And then after the phagosome formation, phagolysosome forms. Then after the formation of the phagosome, what is phagolysosome? Then see here, the lysosome is a vesicle that contains different types of digestive enzymes, especially more specifically the lysis enzymes, okay, lysozyme. Okay, so this lysosome, when it actually when it combines with the phagosome, then we can call it the phagolysosome. Okay, so the lysosomal granules, which exclusively actually discharge their contents into the membrane enclosed vesicle phagosome, and the membranes of the phagosome and lysosome actually then this fuse, resulting in a digestive vacuole. Okay, so then the digestion occurs here. Here, see here, this at the fifth stage, but partially digest the microbes. All here, these microbes, it, mm, uh, and thus after the digestion, these microbes actually they lose the viability. They lose their viability. Okay, so uh, after phagolysosome formation, the first detectable effect on bacterial physiology occurring within a few minutes after engulfment is loss of viability. These bacteria, they lose their viability. So by 10 to 13 minutes after ingestion, many pathogenic and non-pathogenic bacteria are killed, followed by lysis and digestion of the bacteria by lysosomal enzymes. Okay. And lastly, the intracellular digestion here in digestible materials present the residual body of this bacteria. The dead microbes are rapidly degraded in phagolysosomes by various hydrolytic enzymes, including lysozyme, proteases, lipases, nucleases, glycosylases. All these enzymes actually helps to degrade those foreign invaders. Neutrophils die and lyse after extended phagocytosis, killing and digestion of bacterial cells, and this makes up the characteristic properties of pus. Okay, and lastly, these phagocytic cells, they released or discharge of those waste materials as of the actually all of, they discharge of the residual bodies as waste materials. Okay, and these things have no capability to infect, okay, the host body. So now let's talk about the and there are two types of immunity, uh, innate immunity and acquired immunity. Mm, Innate immunity is that occurs naturally as a result of person's genetic constitutions or physiology and does not arise from a previous infection or vaccination is called the innate immunity, and this one is a non-specific immunity. So the innate immunity actually, this one is the inborn immunity. It includes components such as intact skin, salivary enzymes, neutrophils, natural killer cells, complements. Actually, I have already discussed about all these things in our previous lectures. So the innate immune response is activated by chemical properties of the antigen. It is genetically based, and we pass it on to our offspring. That's why this innate immunity is also known as the genetic immunity. Okay. See, here are the characteristics, some characteristics of innate immunity. It recruits immune cells to sites of infection through the production of chemical factors, including specialized chemical mediators called cytokines. Okay, and another thing is it activates the complement cascade to identify bacteria, activate cells, and it also promote the clearance of antibody complexes or dead cells from the body. And it also identify and remove the foreign substances present in organs, tissues, blood, and lymph by specialized white blood cells. And that means the phagocytic cells. Okay. And this thing is also activate the adaptive immune system through a process known as antigen presentation. Okay, it acts as a physical and chemical barrier to infectious agents. So these are all the characteristics of the innate immunity. And now let's talk about the types of the innate immunity. We can divide the innate immunity into three types: species immunity, racial immunity, and the individual immunity. Species immunity is a form of innate immunity and that's shared and limited by all members of a species. Okay, for example, cattle, they suffer from a cowpox virus but appear to have species immunity to the closely related smallpox viruses. Whereas smallpox is a deadly disease to humans, but cowpox is a mild localized skin infection. Okay, that means these two infections, these two diseases are specific to a specific species. Okay, and the another type of immunity, innate immunity, is the racial immunity. Is a form of racial immunity is a form of innate immunity shared by most of the members of genetically related populations. Suppose all the humans, we are same species. We are Homo sapiens, but we have different types of races, like the Asians, Europeans, Americans. Okay, even here the Indian, Bengali people, Japanese. So there are so many races. So and that's why they have and because of their different environment, different habit, different genetic constitutions, their immunity is also different. Okay, so this type of immunity, this type of immune difference is called the racial immunity. Like the northern Europeans appear to be more resistant to the tuberculosis than the most of the African, whereas Africans are naturally resistant to a variety of diseases that readily easily kills those whites means here the Europeans or Americans. Okay, and the third type of innate immunity is the individual immunity. Actually it varies. This type of immunity varies person to person, individual person to person. Individual immunity is a form of innate immunity which is not shared by most of other members of the same race or species. Okay, and because of the genetic variations within the every species, individuals are statistically more resistant to some diseases and more susceptible to other diseases. Like in your family, mm, your your brothers, sisters, or your father and mothers, they have different types of immunity. Some in some cases, uh, when your brother or sister is infected by seasonal flu, uh, you are not infected. So like these variations, these immune variations, some of your family members, they're susceptible to seasonal flu, and some of you are more resistant than others. So this is the scenario because of the individual immunity. Okay, so now let's talk about the another type of immunity, which is known as the acquired immunity. And acquired immunity, uh, mm, actually this type of immunity develops after exposure to a suitable agent, as by an attack of disease or by injection of antigens. And another thing is it is very specific and it also prevents further attacks. Okay, and it lasts for the whole life of the organism, in certain cases for a few years in others. Okay, so that means this type of immunity, they can create memory cells. Okay, that's why they can actually remain within the body for the whole life or a certain period of time. Here are some characteristics of the acquired immunity. First one is the specificity. This immune system has the ability to work against a specific pathogen, not the random pathogen. Okay, and another characteristics is diversity. It also has the ability to recognize a large number of foreign molecules. They are diverse in to recognize they can recognize so many foreign molecules. And the third characteristics is the discrimination between self and non-self. This is very, very important. Um, the immune system does not normally work against the molecules present in the body. Okay, functions against the molecules that are foreign, for example, non-self, okay, any kind of foreign invaders. So at the very beginning, our immunity have to recognize which one is mine and which one is foreign. So that's why, and this capability and this is the characteristics of the acquired immunity. Okay, and the fourth characteristics of the acquired immunity is the memory. The immune system, acquired immune system, they can generate an immune response when the invader or the pathogen attacks the body for the first time. And then after the first attack, they can create memory cell. As a result, when second attack takes place, they can easily identify them and eliminate them very rapidly. Okay. Now let's talk about the types of acquired immunity. There are two types of acquired immunity: active immunity and passive immunity. Okay, active immunity is any immunity that in the body that creates for itself in response to the presence of specific harmful substance, which usually are bacteria or viruses. Okay, so when microbes or parasites attack the body, it becomes active spontaneously and destroy the invaders. Okay. And the active immunity, there are two types on the basis of how immunity is functioned in the body: cellular immunity or cell-mediated immune response system, or the humoral immunity or antibody-mediated immune system. Okay. So in cellular immunity, T cells produce, and T cells, we have already discussed about the T cells. Actually, cellular immunity means the functions of the T cells. Okay. And the humoral immunity, here the humoral immunity means antibody mediated immunity. And you know, the antibodies are actually produced by the plasma cells or plasma B cells. So the functions we have already discussed about the B cells or B lymphocytes are the functions of the humoral immunity. And another type of acquired immunity is the active immunity systems, which is further two types. You know, the active immunity system is another two types on the basis of how immunity is acquired in the body. Okay, naturally acquired active immunity or artificially acquired active immunity. Naturally acquired active immunity, it occurs when the person is exposed to a live pathogen and then disease develops, and then they becomes immune as a result of primary immune response, because immune cells, because the memory cells are produced. So after you have to infected by a pathogen, and then this type of acquired immunity can arise, can form in our body. So like example is like chicken pox or measles. Okay. And another type of acquired immunity is artificially acquired active immunity system. It can be induced by a vaccine, a substance that contains the antigen. A vaccine stimulates a primary response against the antigen without causing the symptoms of the disease. Okay, so thus through vaccination we can get an acquired immune system. Okay, so another type is the passive immunity. So passive immunity means is a type of immunity which acquired by the transfer of antibody from one individual to another. Okay, such as from mother to offspring. And we can divide this passive immunity into two types: the naturally acquired passive immunity, and another one is artificially acquired passive immunity. Okay, so naturally acquired passive immunity, it is occurred when a mother transfer her antibodies to offspring via through their placental route during pregnancy, like immunoglobulin. See here the antibody, immunoglobulin G, immunoglobulin A. These are the antibodies. Those can pass through the placenta and colostrum to the offspring. And artificial acquired immunity, and this is a short-term immunization by the injection of antibodies such as gamma globulin, a type of antibody that are not produced by the recipient cells. Okay, so passive immunity acquired artificially is done by injecting antibodies or sensitized lymphocytes to an organism via vaccination, like when you know, during the corona virus situation, the plasma donation. So plasma donation is a kind of artificially acquired passive immunity. Okay. So these are the differences between innate and acquired immunity. Innate immunity is a genetic or physiological, and acquired immunity induced or pre-existed antibody. It can achieve from this pre-existed antibodies. And innate immunity, it expressed through gene expression actually, and acquired immunity through disease recovery or vaccination. Okay. And response period is within few minutes to hours, and acquired immunity after 5 to 14 days after getting antigens or antibody. Okay. Stability lifelong, innate immunity, because this one is the inborn immunity, and acquired immunity from few days to lifelong. Okay. Some vaccines, they can act for a specific time period, and some vaccines, they can act for a whole life. And memory response, no memory response happen in case of innate immunity, but memory response happen in case of acquired immunity. And the soluble component of the innate immunity, antimicrobial peptide proteins are the soluble components, and in a case of acquired immunity, antibodies are the soluble components. And the cellular components of the innate immunities are the phagocytes, killer cells, natural killer cells, dendritic cells, and in case of acquired immunity, the cellular components are the B lymphocytes and the T lymphocytes. Okay. So I think it's all for today. Thank you all.